everyone, today we're going to be discussing synapsis. Synapsis is the pairing of homologous chromosomes during prophase 1 of meiosis. This is in preparation of crossing over of genes between these chromosomes. Our first example of synapsis is going to involve individual 1 and their 2 chromosomes, as you can see here. This first chromosome in order of genes goes M, L, I, centromere, J, K, H, G. While the second chromosome in order goes gene G, H, I, followed by a centromere, then J, K, L, M. But if we flip this second chromosome, boom, magic flip, now you can see that many of these genes will line up between the 2 chromosomes. But what about these genes in the middle and the centromere? To make these line up, we're going to have to do a loop. More specifically, a pericentric inversion loop, which is what you're seeing here. Pericentric inversion is where one section of a chromosome is taken out, flipped 180 degrees, and then placed back. In this case, for the second chromosome, that would have been the I, centromere, J, and the K. That is why the second chromosome has been twisted, so it can match up with the genes of the first chromosome. To more clearly understand pericentric inversion, I have taken chromosome 1 and 2's genes out of the loop. As you can see indicated by my red underlines, this piece of chromosome 1 was taken out, flipped 180 degrees, and placed into chromosome 2. I'm going to now demonstrate a crossover between the genes J and K that is indicated here with this X. With this crossover, 4 recombinant gametes would be produced. Out of these 4 recombinant gametes, 2 would be normal and 2 would be non-viable. The 2 gametes that would be normal are indicated by my blue marker as well as my green marker. These would be identical to the parent chromatids. My 2 non-viable gametes are indicated by my purple marker here, as you can see, and also by my red marker. These 2 genes would be non-viable because they are missing some genes and have duplicates of others. Now looking at another individual's homologous chromosomes, if you were to look at this first chromosome, it reads MLI, centromere, JKHG. Well, if you look at this second one and read it from right to left, it's the exact same. MLI, centromere, JKHG. No looping or twisting would need to be done to pair this individual's chromosomes up. I'm going to be demonstrating another crossover between genes J and K. Even with the crossover involved, all 4 recombinant gametes would be viable. None of the recombinant gametes have duplicated or missing genes. We can now look at fitness of the individuals, which is the ability of a genotype to survive and reproduce. When comparing the fitness of the 2 individuals in our examples, individual 2 would be more fit than individual 1. Individual 1 only has 2 viable gametes. The other 2 would not survive, while individual 2 has all 4 gametes that are viable and would survive.